Okay, so can everyone see that? Be okay.
Mm-hmm.
As long as you're seeing Boredomresearch, we should be okay. Okay, so I just wanted to say thanks to Computer Arts Society and Sean, obviously, for putting this amazing program together. You know, we've really enjoyed the other presentations, and it's good to be part of this group as well, which seems to be going in a really good direction. It's very fruitful discussions going on. So if you don't know us already, obviously, collectively, our name is Boredomresearch, and we've been practicing together for 20 years now. And I'm Vicky Eisen, this is Paul Smith.
So today we're going to be talking about elephants and peanuts. So apparently, I mean, I didn't know this, but apparently it's a myth that elephants like peanuts. Actually, they're not really that kind of bothered by them. But it's not the only difference that we have with elephants, and we're going to be talking a little bit about the differences that we have with elephants and also the things that can be shared between biomedical science and environmental science, and looking at some of the insights that have crossed those projects that we're working.
We're also hoping to answer the question why COVID-19 might make us think twice about peanut butter on toast. We're going to have an attempt at responding to that. Okay, so we have been privileged collectively to work with some of the world-leading scientists of the world to create expressions of ideas that are deeply meaningful for informing our understanding of the world we live in. So here we are in an A-Life lab in Graz University in Austria, learning about swarm robotics and eating cake, which was very important for our interdisciplinary discussion with the scientists.
So we like to get quite deeply embedded in the science so that we can kind of appreciate both the ideas that the scientists were exploring, but also the methods that they're using to explore them with. And whilst we was in Graz, we built some rubbish robots, and they were rubbish both in the sense that they were made from rubbish, but also rubbish in the sense that they weren't very good. But it was also very much part of how the scientists worked. Even though in Graz they're building some really kind of very sophisticated, robust, highly engineered robots, they often start life as very simple experiments using, quite simple simple electronics and often some household waste as well.
So through this collaboration, we created what you could call a computer simulation. Often we ourselves refer to these as expressions. And this particular work that came out of working with the Graz lab is called Robots in Distress, you can see here. So the artwork consists of emotional robotics, which was also an element that was being explored in the A-Life lab as well.
So, one of the things that we were really kind of interested in here was we were kind of interested in some of the way that science was being used to address problems with this idea of robustness. And what we really kind of felt was that it would be more appropriate to think of ideas of fragility as primary kind of interface, or the primary technological interface between science and ideas of environment.
So these artworks run in real time, and hopefully we can play a little bit of Robots in Distress here. I believe the sound is quite subtle, so you might not actually sound. A lot of our recent works we've produced like kind of video versions of the work, but also we do live generative as well. So they can be screened in film festivals, but the exhibition gallery version is often a real-time generative version. So hopefully, like most of this work, you can kind of see video documentation on our.
So we feel it's important to kind of cross the lines and blur boundaries. There's this kind of, it's kind of interesting that for a lot of human history, there's been very much a lot of importance placed on ideas of specialists and I think it was Robert. Yeah. American science fiction writer stated specialization is for insects or something like that. And so we're really kind of interested in ways of kind of crossing boundaries. For example, here we've got some slides from workshops that we did with animation students, but these were kind of dissection workshops with animation students. So they very much they very much got an opportunity to kind of step outside of their comfort zone and explore kind of creative processes that were beyond their own practice.
So recently, some of the workshops have been like wet lab and dry lab elements as well. So we do these dissecting things, but also we then take those components on screen as well. So you can see on the right that we were doing looking at samples under a USB microscope. We built software for where the people could capture texture from those forms. So the work that we work with the University West of Scotland, these workshops informed our commission that was where we actually created a digital loom that was like a reconstruction of a jacquard loom. And the concept behind this work is that it produces a unique biological form for every human alive at the time of its first launch. So it keeps on churning out 7.4 billion forms.
So here's an example. Here's an example of some of the forms that were produced by the loom. It was one of those things. It's like, when you kind of work, when you've been working with working with the computer for such a long period of time, the loom is very much this thing. It's like kind of part of your heritage. It was the first programmable machine, but also it kind of represents this really kind of interesting point in history where this thing was created that was completely radically new, and it presented these two very kind of interesting opportunities. One was to explore creative diversity, and the other one was to explore mass production. And very much in Paisley, where the loom was used to produce shawls with the Paisley pattern on, which is very well known around the world. This kind of interest in the creative potential of that machine was really strong and was very much part of how it was being used. But but also kind of the wealth that made Paisley a very kind of important place was really the thing that history kind of made a choice, and it kind of chose the kind of the financial, the economic option, or the economic kind of choice out of those two, and kind of the creative potential of that technology became quite a secondary thing. So it was really nice to revisit that and very much think about the creative potential of this machine. And as part of the consequence of that of that industrialization, the pollution from the textile industry made the freshwater pearl mussel locally extinct in that area. So the Paisley forms that our loom creates are very much kind of have this idea of what would a Paisley look like if it was grown inside the shell of a freshwater pearl mussel.
So we create this huge library of forms that we re-stitch together to create this diversity. So another work very quickly that we've produced is Dreams of Mice. And here, like, we sort of, art allows us to understand in completely different ways. And in this work, this is a visual expression that we created of a neural activity of a sleeping mouse. So it's literally a recording of a dream. And this recording was made by our collaborative scientist who's based at Oxford University, called Vad Valiosky, who runs his own lab called BV Lab. And he is a neuroscientist that basically his mum had no idea what that meant. But we were really chuffed because when he actually showed this artwork to his mum, she told him, finally, I understand what it is that you do. we're just waiting for a scientist to reciprocate with us and, you know, like explain to our parents what we do. Sorry, Mum, if you're watching.
Okay. So, okay, so this, so for the last few years, we've been working in collaboration with scientists at Biodesign in Arizona. So with Carla Maley and Athena activists who run 2 labs in Arizona. One looks at cancer and evolution, and the other one looks at cancer, well, looks at cooperation and conflict, and but also kind of thinks about cancer from a psychological perspective.
And they have, based at the Biodesign Institute, have a really fantastic scientific curator who really understands interdisciplinary research. So she's trying to bring, as you know, several artists into that environment to kind of work with scientists, and it's been a really fruitful collaboration, isn't it? 2 years.
Yeah. So in these slides, you can see us kind of standing underneath a cactus with a strange kind of top. This is called a crested cactus, and it's a cactus with a somatic mutation, which in humans is what we might understand as cancer. So it's really kind of interesting that that these kind of mutant kind of mutant cacti are actually kind of very highly regarded, and they have a higher protection status than normal cacti.
This is one of the reasons that we were driven to work with the Biodesign Institute, because we're looking at these images and we're really in awe of them, that they are, you know, they're very beautiful in their form, and you can see that expression of that mutation as a physical form as well.
So back to elephants. So elephants don't like peanuts, or they're not really kind of bothered by them. But a more kind of interesting thing, maybe to do with elephants, is something that's known as Peto's paradox. So when we think of cancer, cancer is something that happens at the level of the cell. So it's really kind of the probability that a cell might develop a mutation that allows it to replicate beyond control. And when you think about that, if you have more cells, then the higher risk you have of cancer. So animals with more cells should have a higher prevalence. There should be a higher prevalence of cancer. But for a while, it's been kind of understood that elephants don't seem to suffer that badly at all from cancer. In fact, they have less cancer prevalence than humans. So, you know, this is really kind of interesting, and to look at kind of nature and to think, well, you know, if there's these kind of really big animals that have lots of cells but they don't suffer from cancer, then maybe there's something interesting going on. Maybe they have a solution. But for anyone that's wondering, it has nothing to do with peanuts. But it does have everything to do with a protein known as P53, or the guardian of the genome. And this, so P53 is produced by the gene TP53, and that's the protein that really does all the error checking. So as soon as a cell becomes damaged in a way that it stops functioning as it should, it's this kind of protein that effectively spots that change and instructs the cells to commit suicide.
So like humans have one copy, whereas elephants have 20 copies. There's, you know, different.
So we've been working with the scientists in Arizona, and their kind of philosophy is really about trying to look to nature to gain understanding, but very much from a biomedical perspective. So their idea that their idea is that kind of nature is this kind of vast catalogue of information, of ways of kind of doing things, and that science can learn a lot from that. And one of the scientists that we've been working with is this kind of remarkable scientist called Dr. Angelo Fortunati, and he is developing new model organisms to try and understand cancer. So there's this idea that in the biological catalogue, there's this huge diversity of life, and that a lot of life may have developed some really interesting ways of defending itself against cancer.
So he's been working with things in the slide, like sea sponges, and more recently these comb jellies, which are really stunning, which we captured lots of video of them whilst we were there, and things like flatworms as well.
So one of the organisms that that he's very kind of interested in, and that we've become quite fascinated in, is this tiny little speck, tiny little blob called a placozoa, and it's really it's the simplest model organism. So I mean, sorry, it's the simplest organism in terms of a multi-celled life. So it only has, I think, about six cells. Five.
Yeah. But there's no reports of cancer and in any other species, well, apart from not the type of flatworm that he's looking at, but in other flatworms. So obviously it's a really valuable species to be looking at in regards to these.
Yeah. But I mean, but he's kind of trying to fix that, because one of the things that part of the research is that he's kind of blasting these creatures with radiation to try and give them cancer, to see what mechanisms they might have to defend themselves. So in the case of the placozoa, they have this really interesting mechanism where they actually reproduce asexually. So rather than, I mean, I think it's believed that they do have sex, but no one's ever seen it. They're obviously very private, very shy creatures. But they get around that by, most of the time, they reproduce asexually. So they literally cleave into two parts, much in the same way if you imagine a cell dividing, or the way that bacteria might kind of divide. And they seem to use this mechanism to cope with cancer. So in the slide here, in the top right-hand corner, there's a dark sort of patch, and that's where the placozoa has been blasted with radiation and it's caused some damage to some cells. And how the placozoa respond is they literally just cleave off these damaged cells. And what happens is that in that cleaving off, you have effectively two placozoa, one that's entirely made up of healthy cells, and one that has a large proportion of damaged cells. And in the one that has a high proportion of damaged cells, you get a kind of a general collapse where the healthy cells eventually die away, and the kind of damaged cells aren't able to perform the functions that would keep the placozoa alive.
What's quite remarkable about this is that Angelo is blasting them. The amount of radiation is in between 80 to 240 grays of radiation. Now, to kind of put that into perspective, if you blast a mouse with 10 rays of radiation, it would die. So this is extreme amount of radiation for an organism, and it's surviving that amount of radiation.
So far, he hasn't been able to give sponges cancer. By the time he increases the radiation to a high enough, the radiation just kind of literally destroys them before there's any kind of noticeable mutation. So very much kind of inspired by Angelo's placozoa, we've been working with him to create a fictional model organism. And this is an organism that's very much inspired by very new ways of thinking about cancer and thinking about cancer treatment. And it's forming part of a short film that's been produced for installation that will have the title In Search of Chemozoa.
So this film imagines a new organism, like we'll describe the chemozoa, and it's presenting a different approach to managing cancer. Not one based on a cure, but a delicate balance. So this film is going to be part of our solo exhibition that was due to be touring this summer, which is titled Restless Balance. And we're hoping that, well, it's just been postponed, so it might be the end of this year.
So here's a few shots from a kind of a sequence in the film where kind of Angelo effectively stars as his imagination, where he kind of he looks through his microscope onto an island And in the kind of seas around this island, there's a rock where this fictional organism can be found. And that rock becomes the home to our in silico model organism that we've been designing with Angela.
So this is the rock.
Oh, so it's here actually.
We have it actually,
yeah.
Somewhere.
So the rock is actually a real rock. We have the rock here in our studio.
So as you can see, it's quite small, but they're very tiny as well, chemozoa, because obviously they've been inspired by placozoa. And so, yeah, we used photogrammetry to just take the images of the rock, the environment at high resolution, but to bring into the game engine.
So our chemozoa, our new model organisms, they live in the holes in this rock, and they have a life cycle where they feed off of toxic algae, and you very much use that to affect their own kind of chemotherapeutic treatment. So whilst we was at Arizona, we spent a lot of time really trying to understand a lot of the research that was going on and trying to understand the mechanics. So we're playing a little kind of clip here from a simulation that we did. This is a type of cancer called Bart's esophagus, and in this form of cancer, it's in the lower part of the throat, and you get these structures, tubular structures, that are very much similar to the structures that you would normally find in your intestines, but they are being kind of formed in the throat, and the process by which they're formed is something to do with the production of kind of mutant cells, and the stem cells generally are located at the base of these structures, and then their kind of mutant clones are pushed or forced up the structure. We was trying to explore some ideas about how that migration might happen, just using physics to kind of, and thinking about the way that the shape of the structure might influence the way that the cells kind of moved. I think it's fair to say it wasn't very well received scientifically. But it did really it did do a lot to help us to start thinking about some of these processes, and start trying to think about the mechanics at the level of the cell.
And that's one thing that we really liked about the placozoa organism, because you can see it as a body. Also, you can see it on a cellular level as well. So for us, it was really important to start doing some of these studies on a cell-based level, because we were interested in the organism being seen, where you could kind of zoom in so you could see it in lots of different levels. So as a complete body of an organism, but also on a cellular.
So we began to build these kind of blobs of cells, and just sort of come up with a base mechanic of cell division, so that the cells would be able to divide and then distribute themselves around a surface, and sort of form a body of sorts. These blobs of cells eventually became creatures. We started to give them the ability to move around and kind of do things, and they became our chemozoa.
Sounds a bit like a pet.
Yeah.
Okay.
So, do you want me to talk?
I can talk about it. Yeah, so this is something that we read about whilst we were at the Biodesign Institute, and it became a very compelling thing. So someone that we've been very influenced by, and you might see that in some of our other kind of creature work, is a biologist called Ernst Haeckel, a German biologist, zoologist, but he discovered a bizarre, tiny, ball-shaped organism that was swimming about in his samples. And it was whilst he was looking for, I believe he was looking for sea sponges off the Norwegian coast, and he just came across one of these organisms. And its cells seemed to possess a strange ability to live independently from the body. So for years, this was thought to be the missing link between multi and singular cell life. But the fascinating thing about this, and when we read about this story, which he named this species the magician's fear, that no one else ever saw this creature. So it almost became like a mythical creature, and a lot of people, some people don't believe in it because they've never seen this creature. But for us, this kind of fictional being was quite compelling, and we were reading about this whilst we were looking. Well, we were in the lab with Angelo, and we were looking at some of these species, and we thought, well, this would be really interesting to create something fictional.
Yeah.
With some of these concepts coming from working with the scientists.
And it's really interesting because, since we've been working on the project, new species of Placozoa have been discovered. So there was originally only one in the phylum, so it was kind of a phylum just with one representative species. But now there's a few more kind of creeping out and being found. But it kind of raised to us the kind of really interesting potential of all the important organisms from a biomedical perspective that haven't yet been discovered. So we were taking some of the ideas that were being explored around different models for therapeutic treatment and thinking about designing an organism that would actually have these mechanisms as part of its life cycle.
So our being sit in this boundary between what is known, and also we are inventing new ways of treating cancer, which we'll go into in a little.
Yeah, so very much so. One of the things that we find really kind of interesting in terms of some of the research coming out of biodesign is it's kind of the way that it's starting to look to I suppose the same kind of understanding that's increasingly becoming important from an environmental perspective. So in agriculture, there's this big problem where our strategy has been to grow our food crops in great big monocultures, and that leaves them very susceptible to insect attack. And our way of dealing with this is just by spraying them with insecticide and just trying to kill everything apart from the plants that we want to eat. And that creates a problem of resistance. That the insecticides, they're very effective in terms of how they kill the weaker insects, but there's always going to be those that have some kind of natural resistance, and they come back, and those populations bounce back, and they become a problem, and they become a problem that there's no way of dealing with. There's no way of kind of managing that problem, and that's very much what some of the kind of scientists at Biodesign feels with the way that it's happening with the way that we approach cancer, that we're just trying to poison the cancerous cells, and that in any tumor there's always going to be a complex mix, a complex diversity of cells, and some clones will be resistant to treatment and some will be susceptible. And what happens is by blasting the tumor with something like chemotherapy, trying to kill the tumor or remove it completely, we inevitably leave behind resistant cells, and it's those cells that then come back and kill the human. So that they believe that there may be there's lots of situations or lots of instances where what would otherwise be a manageable form of cancer is actually being forced or being kind of pushed to a point where it becomes terminal form of cancer. And this is very much kind of taking insights from the way the ecosystems work. That in nature, lots of problems to do with kind of pests and diseases are just managed through complex diversity, and the way that there's a kind of an interweaving of complexity, and certainly in things like permaculture, that those ideas of managing those problems through looking to nature and trying to understand how nature kind of deals with similar problems is leading to different ways of thinking about kind of the way that we grow food crops.
So In Search of Chemozoa presents a fictional model organism that we have created, but our chemozoa have a very high cancer prevalence. but they manage this by poisoning their cells. So they're actually poisoning them with algae. So they can never cure themselves, but they live in a permanent state of restless planet.
Yeah, so because we've created these organisms ourselves, we've kind of forced them to have cancer. They have this cancerous mechanic where their cells become disorderly and would lead to them being unable to survive in their environment. But we also provide an environment where they can then manage that to try and maintain a balanced structure of their cells.
So we're very much still in mid-production. So this is the little teaser, first time we've shown it publicly, of the film that we're creating, which is becoming quite epic. It's a multi-channel video installation. So there's going to be we're working on 3 films for it at the moment.
So
something had changed. There are a great many below the water. Now,
we're having a little trouble with your audio, but I guess we'll watch and maybe it's possible to share.
So I think one vision of the future is that everybody will get cancer at some point in their lives, but you'll just live with it. And so it'll just be a normal part of the stage of life.
Angelo has managed to cultivate some amazing organisms in the lab. They're simple multicellularity. They don't have as many tissues as we do.
How has nature discovered ways of preventing cancer? We translate that into doing better cancer prevention.
Why animals they are not developing cancer, and that they have something that we don't know.
the pale song and we see a song made of a million frail voices united in search.
So we lost a bit of audio there. Is it possible after the talk for us to have a copy of the video and pop it on?
That's kind of slightly pre-released. We've just got a few tweaks that we're going to be making, but what we'll do is as soon as we get a copy finalized for
but it shouldn't be long. There's only a few little bits to do.
I think we got a good impression.
Okay. So as I was saying, they're born with cancer, and the chemozoa manage their mutant cells by moving in and out of toxic areas. They're actually feeding off toxic algae. And as we imagine their world, we learn that balance and stability is won through careful management. And this is taking some of the concepts Paul was talking about in regards to being inspired by nature and the polyglot way of managing these systems. So
very much the way that we've been working is that there we go. So so we built what you might kind of think of as a simulation. So it's a simulation of cancer mechanic built into an organism, and that organism is then able to, or that simulation runs in a game engine, which then allows us to effectively just capture the output from that simulation, but in a more cinematic way. So then we have virtual cameras that we control with my keyboard. So at the moment, I'm having real trouble typing emails because my keyboard has got all these sticky labels on it that allow me to control the camera. And I used to have 2 keyboards, but then when lockdown started, my son wanted the other one to use for gaming, but he also wanted to learn how to touch type. So he kind of sprayed it so that he got rid of all the kind of characters on the keys. So now I have this one keyboard that I'm getting quite good at touch typing now because I'm having to remember where all the keys are. Although I find passwords rather challenging. But yeah, so we have it's a kind of a process that's maybe kind of more similar to kind of documentary film making, where we've created the creature, but then we kind of have to sort of sit and wait their camera to capture it when it's doing something interesting.
We actually realised that we should have invested more time in kind of getting them to be in zones that look aesthetically interesting, because we're like waiting for them to actually move into an area where we can actually capture them, which is quite so like just going a little bit back into like Placozoa. We'll probably have to wrap up quite soon.
How are we doing for time?
I think we've got about 50,
Yeah, you're fine. That's fine. Yeah.
yeah. Okay. So not a lot is known about Placozoa, the organism that Angelo's working. But Angelo has discovered that even though they are very small, they have a huge impact, and obviously they're very valuable for the research that they're undertaking. They can't give them cancer.
But they're also kind of showing that they do seem to play a very big role in terms of balancing their local environment as well. So there's this really kind of interesting idea that they're, and so in our chemozoa, in the creatures that we created, that there's this notion that comes out in the film where, in their attempt to sort of selfishly manage their own health, they're also bringing balance to their environment by kind of consuming toxic algae. So back to peanuts. Right. Peanuts. So right. Why peanuts?
Why are we bringing up peanuts?
Why are we talking about?
It's quite important for now, really.
So elephants don't like peanuts, but humans do. So and one of the things that's kind of really happening at an epic scale is things like peanut butter that contain palm oil, and well, pretty much everything we eat apart from kind of vegetables now seems to have palm oil in it. So in places like Malaysia, there's huge amounts of deforestation that are kind of an expression of this human desire for cheap food oils. And very much so what we have is this stripping away of polycultures and replacing them with vast monocultures of palm oil plantation. And in 2014, we collaborated with a mathematician working in the field of epidemiology called Dr. Paddy Brock, who was interested in how land use change might be behind a rise in zoonotic diseases. And for the few years that we've been showing this project, we've often had to explain what zoonosis is, and but I think pretty much, I think everyone gets it now. So in this case, that the massive kind of pressure of palm oil production is forcing local farmers to have to cut down new areas of rainforest to produce local food. This displaces the monkeys, and then they plant their crops, grow their crops, and the monkeys often return to raid the crops now that they don't have anything else to eat. And that kind of brings monkeys and humans together, and mosquitoes will happily bounce between the two, feeding off of them, and they're not really that picky. But this kind of provides an opportunity for diseases to pass from one species to the next. So we worked with Paddy to build a spatially sensitive model of disease transmission, where from an artistic perspective, we were really interested in what a disease would look like in the landscape. So we found a lot of the models that he was working on really quite abstract, and that they lacked that visual quality that allowed us to really kind of have any sense of what might be going on.
Yeah, and so we were working with light predominantly in this work. So it was so these spiral paths were evocative of the mosquito flight paths, and then the model would go through lots of different stages. So it went through a whole SEIR model, where the stages were from susceptible to exposed to infection, and then ideally recovered. So yeah, so we thought it was a very pertinent work to talk about now in the current landscape that we're in. And as Paul says, that a lot of people are much more familiar with the term zoonosis now in regard to zoonotic infections.
Mm. So and I think both of these works, both the work with an epidemiologist and the work with cancer scientists, they're going to be part of this exhibition that really kind of looks at ways of thinking about human health, but they look at it in terms of, you know, health in the broader sense. So health both in terms of you know, thinking about the way that our cells work, but also health in terms of thinking about us as part of a wider system and how we kind of maybe impact on that system and how that affects our health. And there's these lots of really kind of interesting connections where in very much this research that's thinking about new ways of dealing with the tumor is very much thinking about the tumors as ecologies, and thinking about the best way to manage those is by trying to have the complexity of different mutant clones that then keep the tumor, they keep each other in check. It's a system that's managing itself, and it means that humans can then manage that system in a much more hands-off way. So rather than trying to go in really heavy with some quite nasty drugs, that we kind of ease back a bit and just try and take a much kind of lighter role. And it's really this sort of shared understanding where, you know, it's that same understanding that's very much charged in terms of human health is also quite relevant in terms of how we think about maybe the way that we are interacting with our environment.
So Angelo, who we are working with at Biodesign, recognises that there is much we can learn from species that most of us have never even heard of. And we've had we've had lots of conversations with him about this, including one that we had before COVID-19 kicked off, and where we were talking about species that have been lost and the value of those species in regards to science. So these are not the rhinos and pandas or elephants that we often are fearful of losing, but tiny species of animals, you know, tiny little specks, which many of us are yet to be discovered. Really, no, they haven't been discovered. So I think where we're at. So thank you for your time.
Great.
Very much. Excellent. I had a few audio problems, but hopefully it didn't distract people from the important parts of the talk. I could understand everything, but I just had the odd breakup. We'll see if they're on the recording. I think what we'll do is open it up to questions, but I always get to ask the first question. So I can certainly see the benefits of you as artists working with scientists. It's a very rich vein of ideas and new material. Can you maybe summarize some of the benefits that the scientists maybe report about working with artists?
That's a good question.
I think it's kind of interesting. It's something that we have become aware of, and there is a very kind of different ways of thinking about things from an artistic and a scientific perspective. And I mean, certainly with Biodesign, they're very much aware that all different ways of thinking about some of the issues that they're dealing with have value, and the more that they can kind of talk across disciplines is really useful. I mean, it was interesting that with the mathematical modeler looking at models of malaria, that the first thing we wanted to do is we wanted to see this spatially. We wanted to have some sense of form and structure to that. And that was very much a kind of a concern in epidemiology, that a lot of the models that they were creating were so abstract, and that they felt that they were missing something, and they needed to have that spatial kind of way of thinking about things, but they didn't really have those tools. That wasn't part of the way that they understood and worked with problems. But so so I think they were really interested in kind of seeing how an artist might kind of work with those ideas. I know that when we made that project, the kind of scientists wanted that top-down view, that we actually kind of output the model, because in the work, the camera always looks at the infection from a human perspective. So it's kind of at eye height, and it moves through the landscape and sees the landscape the way a human would. But he kind of wanted to have that kind of overhead, kind of top-down, kind of godlike view of the scenario. So we did output
we did a little mini project for him, so he had his own personal perspective on the infection scenario. But I think with biodesign as well, obviously they're dealing with quite a difficult topic, you know, and it's kind of affected everyone. You know, everyone's been affected by, or knows someone, or maybe been affected by someone with cancer. And it's quite the research that they're doing is quite complex, because they're looking at it from an evolutionary perspective, but also a psychological perspective as well. And I think through the project that we're working on, the multi-channel video that we're working on, it kind of covers a big range of their research, because the Arizona Cancer and Evolution Center is quite big. It's spread over lots of different institutions. It's not just in Arizona. They're working with Bart's Cancer Institute in London as well. So we've kind of taken on a lot of themes from multiple researchers that kind of fed into the work. So it kind of gives it a broader kind of context. Like, you know, like a lot of the research is very focused. You know, a lot of the research working on science research is working on very focused research. So it gives it a more like holistic quality to the different concepts feeding into.
One thing that's happened in quite a few of the labs that we've been based in is you have a mix of researchers, and there's kind of a hierarchy. You know, there's the guy at the top, and then there's the people that maybe haven't published so much, and then there's the kind of, you know, the recent PhD graduates and the PhD students. And this kind of power hierarchy means that there's a lot of people that sit back and they're quite quiet, or you know, that they're quite anxious about how their work's going to be received. And when you kind of introduce artists into that mix, I think it really frees things up, and people feel a lot more comfortable in putting their ideas forward. And in certainly some of the labs that we've worked in, there's been kind of instances where that has brought ideas out that would otherwise have gone, that wouldn't have been kind of brought out, and they've become part of the dialogue in that arena.
And do you as artists get pressure to make sure that your artwork is scientifically valid?
It's not scientifically valid. It's artwork is scientifically invalid. It's you know, it's the one thing. They
don't worry about you stretching stuff and whatever, saying something or doing something that they would say, oh well, it doesn't really work like that.
Yeah, I remember being really scared when we presented Afterglow, because we were part of that was an animate projects commission, and they also organised a symposium. And it was the first talk that we were going to be doing with Paddy alongside us, which was really nice to kind of have the science angle and also the art angle. But Paddy didn't make it because did his wife go into labour? Or like, yeah. And then suddenly we were left on the edge like, oh, we've got to present all the science stuff as well. And it didn't go too badly, because I think someone from the Wellcome Trust was there, and they went, actually, you did a really good job of presenting the science. But I always get really, you know, that we often have conversations with the scientists where they correct you. You know, they kind of like, because they want the facts to obviously be really accurate.
But one of the things, like certainly with Afterglow, that the model itself has lots of things that would make it scientifically weak. You know, in terms of what could be taken from that model from a scientific perspective would be very limited, incredibly limited. But yet, scientific models generally have to have a very let's say a clean way of describing things that allows them to be unpicked and understood in a very precise way, so that you know what's going on. And that means that they seem to lack some of the qualities, some of the noise, some of the complexity that we were able to kind of include from an artistic perspective. So we were able to show the scenario with a huge amount of complexity and it was that complexity that is quite true to maybe how a real scenario would be, that in a scientific instance, they'd have to remove all of that, because otherwise they wouldn't really be able to see what was happening. So there's a very different way that scientists kind of describe things that is scientifically useful, but not necessarily more accurate.
And it's a lot of those little nuances that are actually really inspirational. So like when we're in the lab, it can be just a very flippant remark that scientists projects to us that is like a very throwaway idea. It's almost like trash to them, and we're kind of going, actually, that's really interesting. You know, that's sometimes what we hone in on, the little bits, that the little scraps of bits that perhaps they kind of disregard because maybe they're working on another project or the funding is somewhere else.
And are there any, while we're talking, are there any other people who have questions? You can either type it into the box and I'll ask it, or you can wave and or make a sound and I can bring you in.
Hi, this is Steve Bell. I've got a question.
Hi, Steve.
Hey, lovely stuff. Very beautiful. It's a fascinating talk. And I just wondered whether you could talk, you talked a lot about the science, and I'm interested in what you might say about the aesthetics of it and what it is visually that intrigues you about these phenomena you've been studying.
Yeah, I mean, kind of one of the things that straight away is our organisms are very kind of glowy in a way that that you wouldn't, that is much rarer in the natural world. And in the first iteration of the teaser that we produced, and we kind of showed this when we presented this last time we was in Arizona, there's this kind of line where one of the scientists talks about Angelo and the model organisms he's working on, and we had our organism there, and it kind of it was I don't know what I'm saying, but it was very apparent that there was this kind of strange, like, I wouldn't say awkwardness, but you know, we'd very consciously created this fictional being that was very super everything in terms of its visual presence, but we were trying to kind of create this bridge between that and the science. That I think the difference was quite apparent, and it made it more conscious to us how far we'd kind of pushed things to create this being that was, you know, captivating on screen, but yet it's still trying to engage with ideas that are relevant from a scientific perspective.
Yeah, but sometimes we do feel like we're competing with organisms. I mean, when I saw the cone jellies, they are so stunning. And they'd only recently got them into the lab last time we went. So that was like November last year. And we were like, oh my God, if only they had them earlier, we would have worked with cone jellies, you know? But I think actually we would have really struggled to work with them because they're so captivating in themselves that it's almost like we would have been trying to compete with them.
Yeah, so maybe that's it. It's a competition between us and the natural world where, you know, it's really hard. It's really hard to kind of to imagine things that are in many ways brighter and brilliant, more brilliant than that kind of already exists. And I think we do that by trying to push the visual aspect maybe in a way that maybe that's kind of a part of the fact that we really kind of know that what's happening underneath is actually a lot weaker, really, in terms of the complexity of what's really going on with our organisms. It's so superficial and such a kind of a broad strokes kind of representation of what's really happening that we kind of turn up the brightness a little bit on the way that we express that visually.
I think Afterglow was interesting though, because there's so many unknowns, and so many things that you don't see through the science. And I think that was the trigger for us in that work, because when the scientists were talking about, like, he was saying the last main study on mosquitoes in the 80s, and we were like, wow, you know, like there's not that much to go by. So it kind of gave us a bit of artistic freedom in the sense that we could, you know, really roll that out and let our imagination kind of take over. But at the same time have it driven by those models and that data. I think that kind of answered your question.
No, it is. It's a tricky thing, isn't it? But I just love what you've done with it. Beautiful. Thank you.
Great, thank you.
And Steve, I love your bookcase. Is that real or an overlay one?
I'm afraid it's a real one.
There we go.
Nothing so clever. I did build it myself.
Excellent, yeah. Well, I have a question here from Annie Runkle, who says, can I ask about your name? How come you work under the name Boredom Research? Does boredom play a role in your work?
Yeah, I mean, like, we came up with the name, it's over 20 years ago now, isn't it? And it kind of stuck.
Yeah, you
Yeah.
know, you come up with a name and then you can't really change it. So we kind of regularly sort of kind of re-describe, you know, we kind of post-rationalize what that means. But there's lots of things that I do like about, you know, the idea of boredom and that kind of that creative freedom where you have the opportunity to think about things without the pressure that often exists in a situation where there's an idea that you have to be doing something useful. And it's often in that kind of more free way of thinking about things that that some of the real kind of insights occur. And I think that's probably, you know, I think it really fits the way that we feel that we engage with the scientists now, that we go in and we provide a space where they can play with their own ideas, really. They're playing with their own kind of science, but in a way that is much less pressured and creates a kind of a freedom where some really kind of interesting ideas can grow.
That definitely is something that we take on board in our workshops as well, where we kind of like create spaces where people can effectively just play. And then like kind of things come out of that play that then they kind of get something rewarding out of it, and then they drive the workshop. So we kind of like steer it. But originally we were working under our names and doing much more kind of conceptual kind of art, and Boredom Research became the name, our umbrella name for all our digital art. And then our digital art kind of took off, and we were like, okay, let's just go Boredom Research. We are now Boredom Research. Oh, yeah.
Great. Do we have any more questions? Maybe I might, because we've had an interesting series of 3 talks in this season, and that they've all dealt with different aspects of art inspired by life. And I wonder if because I know both of the previous speakers are here, if either Nicola or Andy wanted to maybe comment on the work from their perspective. I did think the cactus looked like one of Andy's pots, I have to say.
Yeah.
Yeah, I have to admit I was immediately Googling that cactus. That looks fascinating.
Yeah, they are amazing. They look like broccoli, don't they?
Yeah,
like,
it's sort of like this, and that's sort of like, you know, you say like the science behind it is cancerous, but remarkably beautiful. Yeah, that was a trigger for me.
And there's actually quite a few species of cacti that have cancer, and they're very, and you know, they're very kind of highly regarded among cacti collectors, that they're very kind of sought after. Yeah.
Yeah. So the Biodesign, they have their own cacti garden, right? Yeah, cancer cacti.
Right. Yeah.
It's amazing. So it's still in its infancy, but you know, once it grows, you know, it'll be really stunning.
But it's really, I mean, one of the things that is really nice is that it's quite a positive expression of cancer. So, you know, we have, there's so much negativity in terms of what we think about it, but it is just literally a mechanical thing. And in the instance of these kind of these cacti, it's something that is, you know, it's one of the rare occurrences where they're really highly regarded.
Yeah, they are really beautiful.
I think
that sort of perspective, almost saying it's just a differently changed rules of life. It happens to be a rules of life that really negatively impact on us, but it is still just a form of life. Actually, another thing that you were talking about, I think almost like the freedom, I guess, that art gives you when you're working with science to play, which I guess scientists can do it as well, but maybe play in a different way. That's one thing that I've definitely enjoyed, and I've been working, I guess I've done a lot less working directly with scientists as you have, but the few times I have, certainly that's always been an interesting, I think, part of the relationship, and almost the excuse on both sides to say, hey, let's just play.
Yeah, totally.
And I really like, because like occasionally you do come across You know, there's lots of scientists that are also really accomplished artists, but there's lots of scientists that are really kind of keen to engage with art, but maybe aren't so accomplished. And I really like it sometimes when we try and apply ourselves artistically, but we step a little bit too far on the science side and do some really bad science. And it's a kind of a nice reciprocal kind of thing. It's like, you know, okay, so here's some slightly offensive science for you to be polite about.
But Angelo, who we're working with on this project, I mean, he doesn't really see himself as an artist, but he does these amazing drawings of Placozoa, where he puts acetate over the tanks and then draws their movement. And they're really, really beautiful.
I mean, he is actually a really accomplished photographer. He takes lots of kind of photographs. He's very, he takes some really good images of his science. But also, I think that for him, very much, there's a kind of a there's some things that he could do automatically that he does by hand, as in very much the way that when you learn to draw, it's not really about drawing at all. It's more about learning how to see. And he's kind of applying that very much in a science kind of perspective. You know, using the act of drawing as a way of really focusing on the behavior and being very conscious of what he's seeing to help him think about the science.
Great.
Sean, I remember that. Can you hear, Sean?
I can, yeah.
Thank you. In the case of that last remark, I remember when I was closely involved in the Wellcome SciArt program, the scientists used to talk to me about their involvement. I met many of them. And the thing they really enjoyed about the program was they said it brought out the creative side that we thought we'd almost forgotten about. Now, I think in today's way of working, life has moved on and life has changed, and science is recognized as being an extremely creative occupation. But there's something about the process of become a scientist and the peer group pressure that drives out the creativity of quite a lot of scientists, and they were very, very glad to find it again. I think probably, Vicky, you found that in your
I agree totally. That's something that we're very, very aware of, that there's a huge liberation that scientists have working in a more creative way, where, you know, a lot of the structures of science, although that, you know, it's a very creative process, they're quite rigid. And I think just the idea of working, you know, just the idea of thinking about engaging with an activity in something that they can think about in a different way really helps them understand themselves as creators. I mean, we often think about a lot of the models that scientists produce are very much works of fiction. Not all of them like it when we kind of talk about them like that, but, you know, part of the necessity of science is that they have to abstract things, and that abstraction means that they have to find a way of describing ideas that are completely unrelated to what is really happening, but they're still useful. And that's very much a creative process of finding a way of expressing an idea that works in the context that they're working with. And a lot of that, I think, crosses over very, very closely to the way that artists work. You know, that here's something important, but we just need to find a way that we can express that works in a particular context.
I've got a question here, and I feel I ought to, I'll ask Anna's question, then I ought to give Nicola a chance to say something, because I saw that you put your headphones in. So I'll quickly ask Anna Dimitriou's question. Did you find it disturbing or comforting to learn more about the mechanisms of cancer?
Oh, yeah, that's a big question. Yeah, I mean, obviously we're seeing lots of images of tumours and things like that. Are obviously quite disturbing. But I mean, in regards to the mechanisms, we did, I mean, I suppose we're taking it from we're looking at it from an organism kind of perspective, which was something that we wanted to do quite early on, didn't we?
Yeah, I mean, to be completely honest, I think we're very good at, we are very good at sort of just thinking about things kind of detaching and just thinking about the mechanism. It's the mechanisms that are fascinating, and we kind of engage with them in a way that, this is really interesting, this is really beautiful, how can we express that? And that's professionally how we engage with things. I mean, there was some points where I think I found it quite challenging when with certain kind of images that related to forms of cancer that we've had quite close personal kind of contact with. But I do feel that it kind of makes you very kind of, you can't help but it kind of being very conscious of the fact that we all, everyone, you can, when you look at yourself, you're covered in these little mutations, that every little blemish or spot or mark is really kind of, it's this same mechanic of cells being kind of damaged and just behaving slightly differently. And you do become quite aware of that. And I think we have found that a little bit draining to an extent. And we are, we kind of really, we're sort of, I think we are looking forward to a slightly less, a slightly more, to maybe doing something a little bit different for
lighter.
a while. Yeah.
I mean, well, the problem was because we were working with so many different scientists on this project, we kept on coming up with new ideas as well, because we were thinking, we could do something that was a bit more landscape focused, look at the ecology of it a lot more, like from a tumor level. And then we were like, we've been working on this for 2 years. I don't think we can do another cancer project. I'm sorry, guys. We've really enjoyed it.
I think we just need
maybe a little break.
A little break from the cancer would be,
yeah,
probably a good idea.
Okay, and Nicola, did you want to make any connections between your work or have any questions from your perspective?
Yeah, no, it was just a short one. Thanks. It was really interesting. So thank you both. It was the comb jellies, really. It took me back. I don't know if you remember, Sean, when we did Micro World Cardiff. It's, I think, 2014. But Tim and I worked on a comb jelly program, and I'd completely forgotten about it. So thank you for reminding me. I'll have to dig it out. Yeah,
they're
really
so
interesting.
Stunning, aren't they?
Yeah.
They're actually quite hard to capture. Like, there's some footage on Instagram, I think, with Paul filming them under a black hood, because it was like trying to get capture. But we have got some decent capture, and we're going to put it in the film.
I think one of the problems.
Ours was very basic. I mean, it was at the time we were creating all our creatures from body parts. If I remember correctly, it was created from a breast, but the thing that we were particularly interested in was the bioluminescence, and it's actually something that we're wanting to reintroduce into our work. So yeah, thank you.
Yeah,
I'm going to revisit it.
Yeah, they're really compelling, aren't they?
I think when we came out of the lab, we had quite a lot of video of comb jellies and not as much of some of the other stuff that we maybe needed to put together. Things, but
yeah, they're quite hypnotic.
Yeah, for sure. Anyway, lovely to see you again.
And you too. Thank you.
One thing I was thinking, I'm going to sort of direct this to Graham with his EVA, the annual conference hat on, which I think there must be a session on science and art, and maybe art inspired by biology. There's either one session or 2 sessions. We really ought to have a think about that maybe for a future EVA conference.
We could possibly look for some papers that that would cover that area, and then have an afternoon just concentrating on that as a topic. We can certainly write it in as a possible topic for people to write papers on. We can add that as a EVA topic. I think, gosh knows, heaven knows what we'll be doing in November, but certainly, hopefully next July, we'll have our normal papers in the normal way and everything else, and we'll be able to do that.
Very interesting topic to look at.
But it's certainly an interesting area to explore, and it's not one that I think we've had written down as our paper topics up till now. So yeah, it would be all for that idea. 2 or 3 people want to get involved and write us an EVA paper, then it will be peer reviewed by the masses rather than me, but at the same time, you could be in.
Right, well, I think because we've had a really interesting discussion, we've gone on a little bit more than 1 hour, but I think everyone would agree it's worth it. I think before I just wind up, we should say thanks very much to Boredom Research.
Thank you.
Thank you. That's really nice.
That's great, really interesting talk. So like all the others, this will go up onto the CAS YouTube channel and you'll be able to refer to it there. Maybe if you have any additional resources, perhaps the video when you're happy, I'll link that into the materials and I'll share it on the CAS Facebook and website.
Yeah, that would be really good.
So next week is the final sort of full-length talk in this sort of second season of CAS Talks, and I'm going to be talking about the Computer Arts Archive project. Now, some of you might realise, but not necessarily everyone, but all of the artists featured in both seasons of talks all have work in the Computer Arts Archive. And actually, that was my secret criteria for some of the people I asked. And actually, even the second talk on the censor and so on, we're going to be having some of that artist's work in the collection as well. So actually, all of the talks are in the Computer Arts Archive. However, the work at the moment is prints, but that's not the end of where we want to get to. You'll find out next week when I talk about the project. And then the week after, we're going to do the short talks again. And I don't really want to put people on the spot, but Jeff Davis, I think it would be great to have a short talk from you. I can't hear you, but a nod or a shake would be interesting. Yeah, excellent. Yeah, I'm just looking. Graham, you could give us a short talk on EVA. You're muted, but I shall let you have a little think about that.
Yeah, no, it's all right, I'm back now. Yes, maybe we could do that. Sure.
Okay. Terry is already signed up.
Yeah, Sean, I can actually. I'm working on a usability test at the moment. I've been quite busy, but I'll have something to show. We'll talk about maybe some of the old work and then a bit of the new work.
Well, maybe talk a little bit about the older work, because that's going to be an exhibition that we hope to put on in November. So it will give people a little bit of insight into next, exhibition project.
I had an old piece of work in the 1980s called Cow Boil's Head, which was about mad cow disease, which has a biological or disease aspect to it, obviously. So I can maybe talk about that and keep it in this thread of life-related art or death-related art.
Okay, and I can't see if Stephen Bell is still with us. I'm hunting through the screens. But I was going to ask Stephen as well, who was very nice to see him today. But that might still leave us one more slot. And if anybody would like to do a short talk, I'm happy for it to be, anything really. You could be an artist, you could be art historian, writer, talking about a project, or you may not be an artist, but you may have something that you think the CAS audience would be interested in. So people can always drop me a line. And like I say, they're only 10-minute talks really, so not too much pressure. You can float an idea that you maybe haven't talked about before. And that will be the final session, or the final talk in this sort of second series. So next week I'll talk about the archive, and the week after we'll have our short talks. And then I think I mentioned this last time, we'll have a short break, but there will be a season 3. But I am going to focus a little bit on some activities in Leicester. We're doing some digital artwork with local artists, and it's going to involve 3 live events. And I'll invite people to come along if you fancy looking at the live event, finding out about the work produced locally. So for those who don't know, Leicester is practically right in the middle of the UK. It's in the Midlands, the creative naming, regional naming in Britain, the north, the south, the Midlands. And we've got a great local digital arts community. A number of you actually have been to Leicester either to see me or to do work and so on. Hopefully you'll be interested in just catching up with some of the people you may have seen before, or finding out about new people in the area. So I will share the information about those events with everyone, and you're welcome to join us. But I think that is probably everything for today. I think that's been a really interesting talk. So thank you very much again, Boredomresearch, Paul and Vicky. And I can't promise to be as exciting as the previous talks, but next week, hopefully you'll find the talk about the archive interesting as well. Great, thank you all very much.
Thank you.
Bye, and catch up soon, Esther. Great. Okay, see you all. Bye.